In this video, we will cover from the Groups tab to parts of the Statistics tab. As a reminder, unique functionality to iPMart will be covered in detail in this video, and repeat functionality will be summarized with a reference to the appropriate PMart video for more details. Note that all plots are saved as they are made and can be downloaded in the Download tab. See the Download tab video for more information. The Groups tab is used to make sure the sample names match up across the omics datasets and to ensure that groups are properly assigned. If you already have a file that correlates samples and groups, you may upload it here. The file looks like this. If no file is available, here is what the workflow might look like. Under Create a Grouping Structure From, click Data File Column Names. If sample names are kept consistent, then the Auto Find button can be clicked, and sample names can be double-checked. If not, use the I Need to Align Sample Names button and pop up to properly align sample names. After aligning sample names, designate groups in the next tab. Here you can create the new group column from an existing column or manually. Groups can be extracted in two different ways, manually assigning groups or using existing columns. Existing columns can be separated using a common separator in the name such as an underscore or using regex patterns. An example of this is all of the samples of one type ended with a number. We could extract out just letters into a new column labeling the group mock or infection. After specifying groups, we can select main effects and covariates. Main effects are variables with multiple levels that you want to use when comparing the expression of each biomolecule across these levels. When two main effects are specified, the categories from each variable are combined to make groups, and covariates are variables that may influence biomolecule expression, which will be accounted for in the statistical model, but we aren't explicitly interested in quantifying the effect associated with a covariate. Click Verify and use these values. Any samples with NA, those that are not present across all omics types, will be flagged in a pop-up here. These samples will be removed from analysis and integration as we require samples to be present in all omics types to perform integration. Remove the samples and apply the grouping, then continue to the filter step. The next 4 steps are the same as in PMart. Please refer to PMart video tutorials for detailed explanations. The filter step is similar to PMart and will be applied to each omic project separately. Filters remove outlier biomolecules or outlier samples. Note that the filtering option for RNA-seq data are different than the filtering options for MS and NMR. For more details, see the PMart filtering video. Next up is normalization, which is relevant to MS and NMR datasets, and not RNA-seq data, as normalization is done in tandem with statistics for RNA-seq data. Normalization helps adjust omics data to be on the same scale, removing non-biological variations in data caused by instrumentation, instrumentalists, etc. For more details on how to apply normalization to your omics data, see the PMART normalization video. Note that peptide data can be rolled up to the protein level in the next tab, the Rollup tab, which is further explained in the PMART Rollup video. In the EDA tab, users can make various analytical plots including principal component analysis, box plots, and histograms. Widgets under the plots allow for modifying the plot. The Statistics tab contains several statistical techniques and functions. Similar to PMART, the MS and NMR omics, here a G-test for presence-absence and/or an ANOVA for abundances can be run. For RNA-seq data, differential expression can also be calculated. For more details, see the PMART video on statistics. New statistical functionality that is specific to iPMart will be covered in the next video. Perform a statistical analysis for each omic project.